Hello and welcome back to another episode of VCE Biology. I'm Mr. Dean and today we're going to be getting insane in the membrane. Today we're going to be talking all about the plasma membrane. We're going to be starting with a little bit of a cell recap, talking about the structures and functions of the organelles and the typical cells that we talked about in our first episode. We'll then be moving on to an introduction into the plasma membrane, specifically focusing on what is the plasma membrane, where do we find it. Then we'll be moving on to each individual component of the plasma membrane, talking about something that we really do love here at VCE Biology, being each of the component structure and their important function in the plasma membrane. Finally, we'll finish off by putting it all together and talking about the fluid mosaic model and how we're able to bring this understanding of the plasma membrane together under this idea of the fluid mosaic model. Alright, get ready and get excited. Starting with our cell recap, we decided from our first episode that we actually had a myriad of different organelles that exist within the different cells that we have. These organelles that were able to tell us a lot about what kinds of cells we were looking at, whether they were bacterial, plant, animal, archaea perhaps, and we decided that in VCE it's really important to both understand their structure and their function. It was also good to see that a lot of the different organelles we have vary in both structure and function, but they sort of work together in these really sophisticated sort of systems. Looking at that today, for example, we had from our previous episode, we had our rough endoplasmic reticulum that was covered in our ribosomes, and they sort of work together to produce our ribosomes. We then finished the episode by looking at the difference of our eukaryotes and our prokaryotes and seeing how we could define their cells respectively. We came to the conclusion that both our eukaryotes and our prokaryotes differed in two major ways. These were that our eukaryotes had these membrane-bound organelles. Lots of their organelles were sort of packaged in these little membranes surrounded by fluid in the cytosol. And then they also had this thing what we called a true nucleus, or just a main membrane-bound nucleus. So if we haven't seen this word true nucleus, just make sure we understand that it refers to a nucleus that is membrane-bound. Whereas in prokaryotes, most of their organelles are just sort of free floating around in their sort of cell, and that their genetic material isn't covered in any membrane, rather it just sort of exists within its own genetic region. This was an example of one of the cells that we might have been talking about from yesterday, and just recapping that we had a bunch of the different structures that we looked at, and we can see here they do work in these sort of sophisticated systems where we had our nucleus and our nucleolus supplying some of the important genetic information that was important for our endoplasmic reticulum, both the smooth and the rough, that was able to generate some proteins that then worked together with our Golgi apparatus over here that was important in exporting these out of the cell. These also didn't just work in isolation, they required energy, which we got from here, our two mitochondria, and then we can see that this whole cell is working in a really sort of sophisticated system. But that's not what we're here for today. Today we're moving on to something new and something a bit more exciting. We're getting insane in the membrane and talking about the plasma membrane, or the cell membrane as you might read it for in other textbooks or videos perhaps. But as we said in our last episode, we have the plasma membrane or the cell membrane, and it was this structure that we said enclosed the cell. It surrounded it kind of like the walls in our house. And we can also say today we can add this level of sophistication that it separates the internal cytosol fluid, which we can say to be intracellular, from the interstitial fluid that surrounds the cell. So if we imagine a cell existing as it does, we can say that there is fluid on the inside of the cytosol, and we can say that this is intracellular. A way to remember this is that this is in the cell, or it's, out of the cell. It's surrounding the cell. It's what the cell sort of swims in. And that is the extracellular. It has sort of exited the cell. It's sort of outside the cell. And when we're talking about it functionally, it actually has a lot of different jobs that it's working through, rather than some of the other organelles that we've talked about in the past that are sort of important for one or perhaps two different main jobs. The plasma membrane is really involved in a lot of things. It's got its thumb in a lot of pies, as they say. We can say that it's responsible for protecting the cell from its surroundings, as being the structure that encloses it. It's really important to keep the cell sort of safe. It's important in maintaining cell structure and the cell shape, as I just said. So it's really important that the cells themselves aren't just these sort of mushy, sort of gooey things, that they have some sort of shape towards them. It's also important in controlling the movement of substances into and out of the cell, which we mentioned in our first episode, where the plasma membrane is actually able to control how waste is exchanged to a degree and how we can import different nutrients and water itself. Finally, it also facilitates some cell-to-cell communication, which is very important, especially in dividing cells and in understanding what's going on outside the body. If we didn't have this cell-to-cell communication, how does my cell in my blood vessel know what's happening, to the other blood vessels around it, and how does it respond effectively? But of course, where would we be without an excellent picture of our plasma membrane here in a lovely diagram that I've again ripped probably from Google Images. On the side here, you'll see that I've got the four main functions that we described the plasma membrane as having, being it protects it from its surroundings, it maintains cell structure and shape, it controls some of the movement of the substances in or out of the cell, and it facilitates this cell-to-cell communication. The plasma membrane diagram here, I can imagine if you're seeing this for the first time, is actually very complicated. So we've got this really interesting sort of bubbly sort of first layer over here. This is facing the outside of the cell, indicated by the fact the little arrow here says it's the external membrane face. This means it's sort of swimming in the interstitial fluid or the extracellular fluid that's outside of the cell. This similar sort of bubbly sort of surface down here is again touching the fluid actually internal to the cell, so this is touching our cytosol and our cytoplasm, and it's having that sort of contact. In the middle you can see we've got lots going on. We've got these jelly-like sort of arms that are sort of stretching between our little bubbles that are floating here. We've also got lots of different sort of multicolored little structures, being these pink blobby things, these yellow little bally things, and a bunch of the other little sort of aspects of this diagram going on. There's a lot to take in here, and we're going to start by sort of going through each sort of component part by part, hoping and looking at their structure and function so that we can understand what's going on here in the plasma membrane. And the first bit of component that we're going to be talking about of the plasma membrane is going to be called the phospholipid bilayer, actually described from the previous slides as being the bubbly sort of components with the legs shown down here in this little diagram. So the phospholipid bilayer is a key structural component of the membrane, one of the main membrane structures we'll be talking about. It is composed, as its name sort of suggests, as a phosphate head and a lipid tail. We've also got the idea that this is actually a bilayer, so hopefully you guys know from the sort of a word here, we've got bilayer, referring to the fact that they're going to be sort of two parts to this layer, and we can see this down on this diagram. Here we've got our phosphate heads and our lipid tails existing within this little bilayer. So we've got them up here as well as up here, with our little tails meeting each other in the middle. You can see that these two components create what we call this phospholipid bilayer. So we have this bilayer down in this diagram here with our phosphate heads and our lipid tails facing inwards, and then in the reverse we've got our phosphate heads here and our lipid tails facing inwards. So we can say that the components create the phospholipid, this bilayer, with the phosphate heads facing each layer facing either the cytosol or the surrounding interstitial fluid. So basically what we're saying there is our phosphate heads are also always facing the sides that have the fluid facing them. So that could be the extracellular fluid outside of the cell or the intracellular fluid that's inside the cell, with our lipid tails always facing inward and not facing any fluid within them. Taking a deeper look here at the sort of anatomy of our phospholipid bilayer, I've got a diagram here of a sort of single phospholipid, and we've got these first region being the heads. So we've got our phosphate containing heads. These are our heads up here, and they're described as being a very polar substance. So when we're talking about our polar substances, we mean that they are actually able to dissolve in water. The key reason why they're able to dissolve in water is that the phosphate group over here, it actually is a very charged substance. So we've got our negative charge over here on our oxygen, and other parts of our phosphorus as well, being quite a negative sort of part of the phospholipid. Down here you can see that we've actually got our tails, and these are described as non-polar. You'll notice this with a lot of our lipids when we begin to talk about them in a bit more detail, going into our biomacromolecules in a few episodes time, that they're really a non-polar substance. And you can imagine this yourself if you've ever mixed any form of sort of water and oil. You can imagine that our lipids, being these structures, are actually non-polar, so they're not able to dissolve. And so we're introducing two new words here, being that our heads that are polar are what we call hydrophilic. These are hydrophilic, meaning that they actually are able to dissolve in water. And if we think about the sort of breakdown here of hydro and philic, we're talking about them being water attracting. These phosphate heads really love being around some water, and they're happily dissolve in it. Down here, again, we've got our tails, and they are non-polar, and they're hydrophobic. And so if we think about phobia and we think about phobic, these hydrophobic, meaning that they are insoluble in water, and they are water repelling. They don't really like being around water. So our phospholipids, our single phospholipids here, we've got our water-loving heads, our water-hating tails, and sometimes our tails, just to make sure we're clear, can have these little kinks in them, and they can sort of stand out a little bit. This is due to some of the bonding and perhaps a little bit more of the chemistry within the biology unit. When we're talking about our heads and our tails being either able to dissolve and it being able to be not dissolved in water, we're making sure that we're talking about them being soluble in water in particular. That's a really key phrase here. So our hydrophobic tails down here that are insoluble in water, they'd be soluble in other non-polar sort of solvents if we're talking about them, but in water, which is a polar solvent, it actually can't dissolve. So just making sure that we understand what we're talking about when we're talking about insolubility. We're definitely talking about when our solvent is water. But of course, we're never just talking about phospholipids in sort of isolation. We're talking about them, the heads and tails, coming together and forming this double-layered bilayer that we're talking about, and coming together to sort of form this barrier that we can talk about. And this barrier exists between the interstitial fluid that surrounds the cell on the outside, our extracellular fluid, and the internal fluid on the in the cytosol, which we can describe as being intracellular. And so they create this sort of barrier where our hydrophilic polar heads of our tails that are phosphate and that they love being around water, they attract the water, but our non-polar tails form this sort of barrier where they're repelling water as far as they can, and then they're sort of pushing water away, and this creates this really strong little barrier between the two fluids on either side. It's really important to understand that the membrane is impermeable to things that dissolve in water. So for example, thanks to this sort of barrier, we're blocking most of the fluid that exists and around the cell, and by doing so we're blocking a lot of the water as well as some of the ions and dissolved polar molecules. Down here I've just got another representation of some of the polar heads, looking a bit more at our phosphate and our lipid tails, just looking a little bit more chemically. Just to add to the experience of understanding what our phospholipid bilayer looks like so far, I'm going to just show a small clip from a video, which I'll make sure I leave in the description of this one, that just takes a look at a good visual representation of what our phospholipid bilayer looks like so far. So as you can see here, just going back a little bit, we've got our phosphate heads that are facing the fluid on the outside of the cell, and when we look in, we've got this phospholipid bilayer with our phosphate heads and our lipid tails in between, meeting another layer of our lipid tails meeting our phosphate heads. And so we've got this phospholipid bilayer, and as you're going to see, the animation is going to show what happens when we touch it perhaps, and how it may react. We can see that these lipid tails are always repelling the fluid around them. They're very hydrophilic, and they'd hate having any sort of fluid around them, and they really like keeping them to themselves because it sort of guards them and protects them from water. But of course, our phospholipid bilayer and our phospholipid molecules are not the only components of the plasma membrane. Cholesterol is another important sort of part of the plasma membrane. Described here, the cholesterol is the lipid component of the plasma membrane, and it helps keep the membrane fluid. Membrane fluidity is really important to cells all across our body. So it does this in a number of ways, but before we get into that, I want us to look a little bit structurally at what our sort of cholesterol looks like, described down here in these sort of yellow structures. We can see that they again have these sort of long elongated structures, very similar to our lipid tails, and that's because, like our lipid tails, cholesterol is also a lipid itself, meaning that it does add, in fact, some more insolubility to water. So as we said, our lipids are very insoluble. Thinking of our water and oil mixing doesn't really go together, and this happens again with our cholesterol here. So cholesterol is sitting in our plasma membrane, sort of distributed somewhat like this in the diagram, and it's adding a lot of fluidity and increasing the insolubility of our plasma membrane so far. Another important part of cholesterol is adding to the fluidity and restricting the movement of our phospholipids. So as you can imagine, when we've got our phospholipids sort of bouncing up and down as they're attracting and repelling water molecules in the fluid in the extracellular and intracellular environments around them, it's important that they don't get too sort of dispersed around. So our cholesterol here is sort of sitting here, making sure that they're keeping reasonably sort of in time and keeping together as their movement goes around, which is particularly important as the temperature changes. So looking here, we've got when it gets hot, it prevents the phospholipids from being too fluid. So as the temperature increases outside and inside of the cell, our phospholipids can get quite erratic. They can begin to bounce around more as they become more high energy, as the sort of heat around them increases. And it's important to have these cholesterol molecules in here, somewhat restricting the movement. It also is important when the opposite sort of happens. So when it gets too cold, it prevents the phospholipids from packing together too tightly. So as the temperature decreases, our plasma membrane will likely sort of shrink. And then it's important to have these cholesterol components here structurally to make sure that they're not just compacting too sort of closely together, so that they don't get too sort of frigid and fall apart. Just finishing off this session here on cholesterol. Cholesterol, being a lipid, does add another level of insolubility to the membrane, which can be really important, as both parts of the membrane do face a sort of fluid environment in the extracellular fluid on one side and the intracellular fluid on the other. Without the cholesterol in the membrane, the plasma membrane would not actually be as firm and would be soluble to more molecules. So if it's not as firm, it's more likely to be actually sort of crushed and sort of open and spill its contents out into the environment around it. And if it's more soluble to other molecules, more smaller charged molecules like potassium and sodium are actually able to diffuse and come across a lot easier, and this can mess up the sort of cytosol fluid on the other side, because they sort of require a normal or sort of steady concentration of different molecules to be on the inside to sort of function normally. And so if we're too soluble to some of these molecules, they can just come in and sort of interrupt sort of normal cell function. And moving on to the next sort of section here, talking about the plasma membrane, we're going to be talking about some of the embedded membrane proteins that exist within the plasma membrane. So in general, we have two types of proteins that exist within the plasma membrane, and the first we're going to be talking about here are going to be our integral membrane proteins, and these are defined usually as being our sort of permanent proteins. So they're sort of permanent fixtures of the plasma membrane. They may exist on both sides of the membrane, so perhaps they exist on the sort of bilayer side that faces the extracellular, or perhaps they only exist on the intracellular side. And when they do exist on both sides, we can call them these transmembrane proteins. So we'll look at a diagram in the next slide, but these sort of transmembrane proteins just span the entire membrane rather than sort of other integral proteins that may just be on a single side of the bilayer. Functionally, they may actually be involved in a lot of different things. So for example, we'll see in the next slide, but we may have these membrane proteins and these integral proteins that may be acting as sort of transport channels for certain molecules, because it's important that the plasma membrane can control what kind of molecules come in and out of the cell. They may facilitate certain reactions either on the extracellular side or the intracellular side of the bilayer. They may be involved in signal transduction, which is essentially just the communication between the cell and its environment, and they may be involved in some kind of cell-to-cell recognition and cell-to-cell joining. So it's really important, especially when you've got layers and layers of cells, that they join in this really sort of harmonious sort of way. So it's really important that these membrane proteins exist and are able to sort of connect cells to each other. Looking here at a diagram of our integral proteins so far, we can see that we've got a bunch of different examples of these types of integral proteins. So our integral proteins are actually all of these in the example we've got here can also be called transmembrane proteins, because they span the entire length of the membrane. Just to make sure that we're clear, some of the integral membrane proteins may actually only stay on one side, or this side for example, but we can get these transmembrane proteins that go all the way. So here you can see we've got different types of transmembrane proteins and integral membrane proteins that may exist in the plasma membrane and what they do. So we might have some that act as a receptor for particular, say, cell signaling or cell communication. We may have some that act as an enzyme and facilitate reactions that occur in the intracellular space or the intracellular fluid. We may have some that are transporters, so they can either transport things in and out, perhaps they need to change shape, perhaps to be able to get certain molecules in and out, or we might just have ion channels like these that allow ions to sort of flow simply in or out of the cell. Moving on to our next family of membrane-bound proteins, we've got these things that we can call peripheral proteins. And peripheral proteins, as opposed to our integral proteins, aren't actually permanent members of the plasma membrane, and they're not exactly embedded within it. So what we mean by that is they are actually sort of external to the membrane itself, but they attach to sort of other parts of the membrane bilayer, and we'll see this on the next sort of diagram. Instead, that we can say that they attach to the phospholipid bilayer or to integral membrane proteins on the inside. So we've got these integral membrane proteins in the plasma membrane and the phospholipids that make up the plasma membrane, and then we've got these peripheral proteins that sort of exist and attach to it externally. Functionally, when we're talking about peripheral proteins, we're talking about them being involved in facilitating reactions, just as the enzyme protein that we saw in the previous diagram, signal transduction and communication, as we saw in the receptor molecule in the membrane protein on the side before, and again, that sort of cell-to-cell recognition and joining. Here is our diagram of our peripheral proteins we've got here, and as you can see, just as said on the previous slide, our peripheral proteins being this D, E, and C molecule, they don't exactly sit within the membrane itself, but they sort of attach to structures. So here they're attaching to two different integral proteins, both of these being transmembrane proteins, and then we've got our E and C membranes actually being attached to the plasma membrane bilayer itself. And so they are sort of membrane-bound structures, sort of, but remember that they're not permanent and they don't exactly sit within it, sort of more on top or beyond. Finally, finishing off our sort of family of all the different sort of parts of our cell membrane, here we've got our membrane carbohydrates. Now, membrane carbohydrates are reasonably simple. So we've got here that our carbohydrates is our sort of sugar molecules that exist within cells. They attach to proteins or lipids to form these things that we can call glycolipids or glycoproteins. We'll see this on a few different sort of slides and diagrams that I've got down here. But these glycoproteins and these glycolipids, I should say, similar to sort of our peripheral proteins, they attach to sort of the embedded surfaces that are already within the membrane. And you can see them down here. So we've got these probably our proteins down here, probably an integral protein down here, and we've got our carbohydrate portion, and together we call this a glycoprotein. So we've got our glyco being similar to our carbohydrates and being synonymous with our sugars, and then we've got our protein part, and I'll just quickly find over. And so over here we've got our lipid portion here, and we've got our carbohydrate portion, and then we've got over here our glycolipid overall. And so what are they involved with, and what is their function now that we've just observed their structure? Well, they're involved in cell communication and adhesion of hormones and antibodies to the cell. So what do we mean by that? We're basically saying that these glycoproteins and these glycolipids are really important for cells to be able to talk to each other, and they're really important in enabling hormones and antibodies in other cells to be able to attach to a cell to be able to do their job. Hormones so that they can then act on the cell and induce an effect, and antibodies to help in perhaps an immune response. Over here I've just got another sort of example of how plasma membrane might be drawn, and again we've got our sugar portion showing again what our likely our glycoproteins will look like. And just looking over here, we've also got some, I can't quite see them here, but over here we've got some more sort of sugar structures, and perhaps we haven't got any examples here of a glycolipid, but just making sure that our membrane carbohydrates, they come into these sort of two pairs of being a glycoprotein and a glycolipid, and they sort of sound like what they are. They are carbohydrates attached to either proteins, or they're carbohydrates attached to lipids. They're involved in cell communication, and they're involved in sort of cell-to-cell and molecule adhesion. And so putting this all together, now we're going to look at our fluid mosaic model. So our fluid mosaic model was actually sort of proposed by S.J. Singer and G.L. Nicholson from my notes down here, and it's sort of a description of how the plasma membrane sort of occurs. So looking at our notes that we've seen thus far, as we've looked over all the families of different molecules that exist and sort of compose the plasma membrane, so we can say that the plasma membrane obviously contains many different parts, and that these components come together when we discuss the fluid mosaic model. So the fluid mosaic model, as we talk about it, it sort of has these sort of two major sort of themes and these two sort of ideas that they come together. And the first one is that the fluid in the fluid mosaic model sort of represents the idea that the membrane itself is fluid. It's fluid, it's dynamic, it's flexible, and it's definitely not by any stretch of the imagination something we can consider to be turgid or stiff. So it's definitely got this sort of really dynamic and fluid by nature. It's got this sort of defined shape, sort of perhaps think of blocks of jelly. So they've definitely got a sort of defined shape, but they're not sort of structurally like super sound or sort of stiff by any means. The second part of the fluid mosaic model is going to be the idea that it's a mosaic. So when we talk about the membrane as a mosaic, we mean that it's composed of many different sort of different parts, and that these contribute to its overall function. It's also important to understand that the fluid mosaic model's representation of the plasma membrane also suggests that the parts themselves aren't exactly fixed. So when we're talking about them being fluid and dynamic, we're not just saying that the integral proteins themselves always sit exactly where they are, or that the peripheral proteins always sit where they are, or that the glycolipids and glycoproteins always exist with are. So being dynamic and being within the plasma membrane themselves, they're sort of in a constant state of motion as they interact with the two different sort of extracellular and intracellular spaces. And just to finish up our second episode of VCE Biology here with me, Mr. Dean, we're just going to have one final look at the video that I mentioned earlier that will be down in the description, having a look at the fluid mosaic model sort of in its entirety. So again, looking at this really fluid mosaic model where we've got lots of different parts, and they're not really sort of tightly or structured very rigidly. So we've got our plasma membrane here with our lipid bilayer, with our phosphate heads and our lipid tails, and then we've got all these sort of other structures that are beginning to come into existence. So we've got our transmembrane integral proteins, we've got some of our other sort of peripheral proteins sort of coming in around, we've got different things attaching, we've got some ions and things sort of floating through, we've got some things sort of bouncing back because we understand that it is a semi-permeable membrane, we've got different sort of pores and things happening, probably another sort of integral protein, perhaps a transmembrane protein if it's going all the way through. And again, just reiterating that it isn't a sort of static structure, it's definitely a sort of fluid structure that's sort of in this ever-casing movement, and these structures can sort of move around. Hope you've enjoyed our second episode of VCE Biology. Again, let me know if there's any recommendations you'd like to make.